Okay, so I'm going to go through a couple of things today for the neuromolecular biology that relates to behavior. And you need to be sure that you understand how these different techniques work. We started off with a number of basic techniques, and now we're going to use them in a more refined manner. And the refined manner is what I'm going to be asking you to know in a little bit more detail. We have talked about in the last lecture about the Cree-Lock system. And the Cree-Lock system is really powerful because it allows the spatial expression of different types of genes. However, you're going to see that there's a variation of this that's very commonly used now, but you need to know a little bit of neuroanatomy in order to be able to use this new variant of the Cree-Lock system that I'll be talking about today. So we have looked at one system where you have a TAH positive mouse that contains Cree recombinase in every TH positive neuron. But now you're going to see something a little bit different in today's lecture where Cree recombinase is actually going to be packaged in a different way. So be on the lookout for that. We'll go back over optogenetics as well as pharmacogenetics, and I'll give you an overview of what the differences are. You'll see these a lot in papers nowadays. Optogenetics is still the dominant technique that's used by most labs, and in fact, you're probably not going to see a lot of people using pharmacogenetics in the next few years, but it's an important technique to know because it's still in use. If we have time, and it depends on how quickly I can get through slides, I'm going to introduce you to this inducible diphtherotoxin receptor mouse, which is really important in neurobiology, especially when we start talking about things related to how we can look at fear memory. And then I'll give you a quick overview, if we have time, into the remainder of the course as well. And all of this is important for you. You should make sure that you know it. It shouldn't be anything where you don't understand it. If you don't, just jot down what you don't understand and come and see me. I don't want you to be telling me that I wasn't really clear on whether it was this system or that system, whether it's white or gold or blue or black. Should be no questions whatsoever. You should know 100% like what is going on in these systems. We talked about optogenetics, and I had mentioned that there were a number of variants for optogenetics, that you can turn on neurons that express channel rhodopsins. These are non-specific cation channels, which is why you see the influx of things like sodium, but I also told you that calcium will enter as well as potassium leaving. It's not shown on this diagram. We haven't really talked about the halorhodopsins, but we'll start talking about the halorhodopsins today. If you want to inhibit neurons, you overexpress these halorhodopsins, shine a different color light on them, and halorhodopsins work. All of these up here are channel rhodopsins. They're all derived from the opsin protein that are found in algae. A newer variant of all of this is known as OptoXR, and this is a G protein coupled receptor that can potentially get turned on by light. It's still in its developmental phase, but you'll see why that might be important, especially when we get into pharmacogenetics a little bit later on. And this OptoXR system, where you might be activating G proteins with light, is sort of the next variation of it. You do not have to memorize all of the differences down here. It's just to give you an overview that some things are really fast excitation. Some things are really fast inhibition, the halorhodopsins, and we won't really be talking about this system at all in this course. It's another option available for you, as well as the OptoXR system, just as a comparison. Any other questions before we move on? Hopefully you'll know that the channel rhodopsins are excitatory. There were a number of papers that came out. These two seminal papers that I mentioned at the end of the last lecture, two seminal papers back to back, same issue of Nature Neuroscience, both using optogenetics, both targeting the same area of the brain, that were published in early 2013. Landmark papers, because two groups coming up with a singular hypothesis is very unusual. Carl Diceroth, for example, is actually found on both papers, one as an author and one as a senior author. And this optogenetic method really helped to elucidate the mechanisms of this firing rate pattern that was somewhat mysterious. No one really understood, like how could the changes in a firing rate in the VTA to nucleus accumbens, if you increase the firing rate, why are you more likely to become depressed? Isn't the VTA nucleus accumbens normally that's something that is a reward pathway? Like you all learned that in your second year classes. Why is it possible to have a change in firing rate in the reward pathway that eventually leads to depression. And again, all of this was done back to back, almost at the same time. This paper that I'm going to be unwrapping for you was actually received a little bit earlier than the paper that was submitted by Carl Diceros' group that was looking almost at the same thing. All of them got published together, and you can see that from the time that it was actually received, all of the follow-up experiments took several months for them to actually do, and some of that gets back into different types of vectors that had to be created. I had given all of this to you before, just as a reminder. In this particular system, this was the original construct that was used in that first paper that I showed you in that previous slide. In this particular paper by Dipesh Chowdhury out of Minghu Han's lab, this is the same construct that he was using initially to do the characterization. So he was looking at this channel rhodopsin, excitatory. You could see if it was being expressed because it's linked to this enhanced yellow fluorescent protein. Wherever you see enhanced yellow fluorescent protein, you get channel rhodopsin, okay? And we talked about how it could be specifically targeted to Cre recombinase containing tyrosine hydroxylase neurons, dopaminergic neurons within the VTA. He does exactly the same thing that Carl Diceros starts off with. So he also, because it's the same mouse system, it's the same sorts of things that I showed you before, he wanted to know, if I inject this virus that I just showed you on that previous slide into the ventral tegmental area that's full of Th positive neurons in red that you see here, if you take the ventral tegmental area out, stain it for tyrosine hydroxylase, you see all of this. If I go in and I start looking at the expression of the virus, where do I see the expression of the virus? And again, it's really in the Th positive neurons that he sees channel rhodopsin. Wherever you see overlap, this overlap is there, and about 60%, not quite as high as the original paper I showed you, but somewhere between 60 and 70% of the neurons that were Th positive were also expressing this construct that he was able to put in. Just a couple of really quick things. I mentioned at the end of the last lecture that if you're doing that social stress model where you have an aggressor mouse and you're put in with an aggressor mouse like a bully, in order for you to show signs of depression, it usually takes about 10 days. And again, keep all of this in mind. This social defeat system is a 10-day model system. However, using optogenetics, you can actually speed this process up quite a lot. The one thing that I told you, sort of as a background, was that there was a change in the firing rate that had been observed prior to this paper. Many individuals, including one of the senior authors that I showed you on the two previous slides, or the slides previously, one of them was Eric Nestler. So Eric Nestler had proposed, you increase the firing rate, you increase depression following social defeat. If you decrease the firing rate or have it stable, then you don't have any social defeat, you don't have depression, or sorry, following the social interaction, you don't have depression. So this paper, using optogenetics, wanted to know, can we actually prove that that is the case? Can we induce depression by changing the firing rate of this group of neurons? And that is the basis of this paper. Other papers had looked at the change after depression. This group wants to know, can I induce depression by changing the firing rate? And there are two different types of firing. One is known as a tonic firing. It's a consistent, continual firing that is generated. So you stimulate your channel rhodopsin, you flash on blue light, and when you flash on blue light, you can have 0.5 hertz stimulation. And you can also have another pattern, which is known as phasic. So phasic is in bursts. So you see bursts of action potentials, which occur at like 20 hertz intervals. Importantly, they wanted to keep this very, very consistent. So they had 5 spikes for each 10-second interval. So in this 10-second interval, you'll see 5 action potentials being generated. So 5 action potentials over 10 seconds equals 0.5 hertz, right? If you do the division. And in the same manner, before you start the next 10-second interval, you have it all at the very beginning, then you let it rest, and then you start your next 10-second interval, and this wouldn't be included. So only this burst is included. So in other words, it's the same number of action potentials, it's just delivered at a different frequency. Is that okay for everyone? Okay, so we have the same sort of spiking activity. we have phasic versus tonic. And again, in this particular model, they're looking at how much time do you spend in the interaction zone after you have a change in firing rate, either tonic or phasic, and how much time do you have with this target animal in this particular zone. So if they're depressed, they will spend less time with the animal, which is what you see here. If you have phasic activation of the VTA, the animal actually spends less time, they're less curious, they're less likely to interact with the animal in the interaction zone. In addition to that, they also show that there is a decreased sucrose preference. So tonic doesn't have any effect. A control vector, the enhanced yellow fluorescent protein by itself, and phasic activation has no effect. It's only if you have channel rhodopsin and you can stimulate bursts of activity in a phasic manner, it's only this that causes this lack of interaction, depression, as well as the lack of sucrose preference, anhedonia, as part of depression. Okay, so it was a really elegant experiment that was done to show that even optogenetically, by changing the firing rate pattern as a proof of concept, you could actually induce depression. And again, they're measuring in that previous panel, what they're measuring here is if you are depressed, you are less likely, following this sort of social defeat, after 10 days you would see this, or after optogenetics you would see this. If this is the interaction zone, they don't really interact at all. They get to see where the mouse is. And when they see where the mouse is, a normal animal who's not depressed or has not had social defeat leading to depression over 10 days, or has not had phasic activation of the channel rhodopsin, will spend a lot of time in this interaction zone. However, after phasic activation, you don't see that type of interaction at all. It goes down dramatically, and the same thing with sucrose. So just by changing the firing rate using channel rhodopsin, you're able to do this. And Chowdhury's paper was the first to actually go back and show that all of the original observations that were done by Eric Nestler's group were indeed accurate. By changing the firing rate, increasing action potentials, you could actually change behavior, which is what is being shown here, all through the VTA, only in VTA neurons, and again, a really important paper. Okay, so we won't go into this in a lot of detail. There are a couple of really important things that were brought up in this paper. It's most likely if you have these mice, and it had been known for a long time, if you have mice and you engage them in social defeat, or you hang them upside down by their tail, not all the mice respond in the same way. Some mice are resilient, some mice are fall easily into depression, but not all mice, even though they're genetically identical, fall into depression in the same way. They don't have that same overall change. So if you just give them a sub-threshold model, and we won't go into the details of this sub-threshold social stress. It's like having a bully, but the bully is kind of like your friend once in a while. So this sort of sub-threshold, that bully doesn't bother me all the time type of paradigm, they don't really exhibit any social avoidance or depression-like behavior. However, one of the things that they found was that following this initial sub-threshold stress, or the sub-threshold model, where they're interacting, but the interaction is mediated so they don't feel like they're vulnerable, they don't feel like they're being bullied. They are though vulnerable for more depression if they have increased stress later on. So it's like you're priming the system. You're getting them ready to have depression. That particular scenario, that particular phenomenon also hadn't been shown. And it's important because remember, one of the things, just to wrap up depression, one of the things that we had sort of talked about is we don't really understand depression in the truest sense of neurobiology. Secondly, we know that if you have a bout of depression, that you're more likely to fall into depression subsequently. And if this model sets up that there is a certain vulnerability that you can elicit, that might help us to understand and tie up some of these loose ends. So if you go back and you start stimulating the VTA dopaminergic neurons of those animals that had just been under having depression. So they don't exhibit depression yet. They don't have any of the problems in social interaction. They still interact normally. They still have sucrose preference. So they'll drink as much sucrose water as a non-stressed animal. But if you take those, the VTA neurons of those sub-threshold stimulated mice, and you use these optogenetics, what they see is that they are a lot more susceptible, and they will fall into depression fairly easily compared to other animals. So again, just to sort of wrap a couple of things up, the main point of all of these different types of experiments was that if you have phasic stimulation, bursts of action potentials, increased action potentials, in other words, they will have a robust increase in this depression-like phenotype. You can measure it through interaction or through sucrose preference as anhedonia. And again, compared to their control mice where they have EYFP but no rhodopsin associated with it. When you shine light on it, these don't activate at all. So it's a good control in terms of phasic stimulated mice. So this confirms this functional importance of phasic firing pattern differences in the VTA to nucleus accumbens that during exposure to stress will lead to things like depression, at least animal models for depression, whether it's interactivity or sucrose preference. So again, the really, really cool thing is that just by changing the firing rate, you don't have to take an animal through 10 days of this model. 1 day is enough to actually induce this. One bout of this phasic stimulation is enough to cause the same sort of phenotype that we looked at earlier on. We don't see this phenotype in tonic stimulated mice where you have a consistent, constant rate of action potential firing, or in the EYFP mice. Okay, so we can induce these depression-like behaviors really rapidly. All perfectly clear, right? Simple change in firing in the VTA, in those dopaminergic neurons, is enough to give you the same depression model as if you took mice through 10 days of social defeat stress. Clear, right? Hopefully. Any questions at all before I move on? All using optogenetics, yes.
So for how long were they changing this firing rate?
Oh, okay. So is the change that they induced long-lasting? It turns out, and I think I have a slide coming up, that it is. So once you entrain these VTA neurons, it turns out that if you have phasic firing, they become entrained to phasic fire continually. So it was enough to actually do this. Okay. Yeah.
Can you, like, I guess, make them not depressed, or I don't know, like treat their depression by like weaning them off phasic firing?
Yes. Yeah, I'm going to show you that in a second. So that's the other second half of this. So this by itself would have been probably good enough for like a lower impact paper, maybe like PNAS or others. But since it's his nature, they had to do some other things to be able to publish this, and I'll get into that. So if you stop this phasic firing, can you actually reverse the phenotype? And they do exactly that. Yes, you had a question at the back. I'm not going to, yeah, so I'm not going to get to all of the different aspects of the social interaction, but one of the things that you want to be able to do is have a target animal present, or you want to just see how it's moving around in the box. And that's sort of the target, no target. We won't worry about that. I just showed you the target interaction, which is the indicator for depression. So one of the things that was really surprising about this paper, you needed a stress. You could have a sub-threshold stress. You could have a mouse that had other things going on, but you needed some sort of stress. So sub-threshold stress, you put in and you cause phasic firing. They become depressed after like one single bout of phasic firing. However, in naive animals, so they've never been exposed to stress, they've never had a bully interacting with them. Totally naive animals, change in phasic firing isn't enough to do it. Only subthreshold, where you don't see depression yet, but then you induce optogenetic firing, then you see that change that I just showed you. Naive animals had no effect. So in other words, it goes back to our original model, right? So you don't spontaneously fall into depression, right? You can't be walking down the street and suddenly be depressed. You need some sort of stressor in your life. That stressor might or might not cause depression. And what they showed was that sub-threshold paradigms where you don't have the end outcome looking like depression, those animals are okay, they look okay, but then you put in channel rhodopsin, you give them one bout of phasic firing, they become depressed. You still need a stressor in order to cause this to work. A naive animal has no effect. You can't change the firing in a non-stressed animal and have a depression-like phenotype occurring. So again, there seems to be a lot of interest into how the changes of chronic stress and different types of stress increase or inhibit the activity of these VTA dopaminergic neurons. And this is sort of the next item that they try to address in this. So you need stress in the VTA in order to elicit this sort of paradigm that I just showed you earlier on. And again, we've talked about this a number of different times. Chronic mild stress, chronic social defeat stress actually are not equal. So they will have different changes in the extracellular level of different neurotransmitters, different brain regions seem to occur. And so in the social defeat model that they're looking at, it seems to be very, very specific. One of the things that you were asking, so just in this sub-threshold mouse that I've just stimulated once, is this a long-lasting change? It is. This is a long-lasting mechanistic change. So if I go in these sub-threshold mice that don't have any signs and symptoms of depression at all, and then in those sub-threshold mice, I push them over the edge by giving them phasic stimulation, they become depressed. And these changes are actually very, very long-lasting and actually occur at the level of action potential generation. So if you look, if you just have EYFP and you look at the normal firing, oh, and by the way, before I go on, I know it's a little confusing. In E, this goes back to the original papers. And in E, what you're seeing is sort of a recapitulation. If you take a socially defeated mouse that has had any kind of social defeat and it has channel rhodopsin, and then you take out the ventral tegmental area and you look at the neuron firing pattern, these are the firing patterns that you would normally see. This isn't the induction of this isn't the induction of the firing pattern. This is the normal firing pattern that you would see after tonic stimulation or after phasic stimulation, okay? Does that make sense to everyone? You are not inducing these changes. This is what happens after tonic stimulation has occurred. This is the normal firing pattern in the VTA after that firing pattern change has been elicited. After you've had like subthreshold social stress, you've done phasic firing, you take out the VTA and you look, what is the normal firing pattern like now in the VTA? This is what happens. They've increased their action potential generation, right? Is everyone able to see that you get much more action potentials here on the right than you do under tonic or under control vector infection? Yes? No? Okay, great. Again, the firing rate goes up and you can actually measure this. Does anyone know what the Rio base or picoamps actually shows here? Let's go back to your classic electrophysiology. If you're measuring from a neuron and you are looking at the amount of current that you have to inject in order to elicit a set level of threshold activity, what does this indicate? Everyone's kind of looking at me like, I have no idea. Any guesses? This is really important, yes. But what does that mean, sensitivity? Okay, kind of. Do you want to follow up on?
Just guessing.
Yeah, yeah.
Maybe it's the how close it is to already firing action potential, like where its resting membrane sits at is a little bit higher up than.
Yep. Okay. Absolutely. So, is it easier to do this in just the EYFP? Is it easier to stimulate them with tonic, or is it easier to stimulate them with channel rhodopsin after phasic activation? After phasic, I only have to inject about 15 picoamps of current to be able to elicit activity in these groups of neurons. So they are much more excitable, and they fire many more action potentials depending on how much more current I inject. So compared to like tonic stimulation, not only are they more excitable, so they're more excitable, and this Rio-based measurement tells you, you can actually see how much current do I have to inject in order to generate an action potential. It's much lower after phasic stimulation. So after phasic stimulation in these animals, they are much more likely to start firing action potentials, in other words. And when you start firing action potentials, they fire many more action potentials. The spike number actually is significantly increased at every level that you look at. So they're easier to excite, and they fire more action potentials. Exactly what Eric Nestler had hypothesized back in 2008 and 2009. Using optogenetics, using subthreshold, normal-looking mice that you push over the edge with one bout of tonic, or sorry, one bout of phasic, and as a control tonic, you're able to show that these are long-lasting changes. And so it was a really cool idea that you could actually have a low level of stress. One more stress is enough to push you over the edge into depression that will then result in long-lasting changes in the firing of action potentials in the VTA. Exactly as hypothesized. Any questions before we move on? Yes.
I'm not really sure whether I get this, but like you add the basic stimulus, and then the tonic is like because this activity at a
So
level.
we're looking at 3 different mice here on E. So we're looking at EYFP. So EYFP, they don't have channel rhodopsin. It's just the normal control vector. You want to see if the actual infectivity does anything to your neurons. If you take channel rhodopsin plus EYFP, and then you do tonic stimulation, and then you go back in after you've done tonic stimulation on subthreshold stimulated mice, this is the action potential firing rate that you see under normal conditions. And this is phasic. After phasic stimulation of those subthreshold mice that don't have stress, this is the increase that you see. Okay? And they definitely go up in rate. Any other questions before we go on? So your level of stress, and one of the reasons why many individuals are really interested in your level of stress, many of you have different set points for your stress level. Some of you might be able to handle having a part-time job, going to school, managing someone in your family, many other things, and you'll be able to handle that fairly well. And that chronic level of stress all the time might not even approach this. However, what this suggests is that if there's an extra stress that pushes you over the edge that will increase the firing rate in your VTA, it might lead to things like depression. At least in this social defeat model, it might not be a universal model. So stress is actually really important. So I'm going to take a little bit of a detour right now. This is a pure molecular cellular neurobiological technique that's used everywhere. And I need you to understand it because of the next sets of experiments that are done in this Chowdhury paper. I don't know what everyone's background is like, and so I need to make sure that you are able to follow this along. We know, for example, that the VTA sends its axons and makes synapses to another group of neurons in another structurally different part of the brain, right? What area of the brain does it make synaptic connections to? Nucleus accumbens is one. Where else does it make connections to? Prefrontal cortex is another area, and there's actually a third area that we talked about as well. The habenula is another area that has projections coming in from the the nucleus, from the VTA. And so one of the things that you do is, if you know of the target group of neurons, for example, the nucleus accumbens, or the habenula, or the medial prefrontal cortex, you can actually put in a dye, or you can put in a viral subunit toxin, like or a bacterial subunit, like cholera toxin B subunit. The neurons will take that up wherever you inject it, and then it will actually allow you to see if you are actually making a connection between these two different areas. It will be retrogradely transported back to where the original neurons are coming from. So if I thought, for example, I think the nucleus accumbens is actually the target of these VTA neurons. I'm not sure. If I inject a dye into the nucleus accumbens and it gets taken up by the VTA neurons, I should be able to see the dye in the VTA neurons. And this is occurring through retrograde transport. It gets taken up back along the axons, back to the cell bodies. I would see this dye in a group of neurons, for example, within the VTA, if I injected it into the nucleus accumbens. Is that okay for everyone? So it's a retrograde tracer. There are many types of tracers. This is a retrograde tracer. You put it into the target, it gets taken back to the origin of the axonal targets. So you can also use viruses. I told you earlier on, much earlier on, when we started talking about depression, that neurons are actually, that viruses are actually neurotropic. Viruses like infecting neurons, and viruses can be transported along neurons. And I just told you about retrograde transport. You can put cholera toxin B subunit into the nucleus accumbens. You will then have it traced all the way back to the VTA, because cholera toxin B is retrogradely transported back to the to the VTA. And that's one type of retrograde tracer. It is a chemical or a pseudochemical like tracer. I told you that viruses can do the same things. If I had pseudo-rabies virus, or PRV, PRV does the same types of things. If I inject it in the target, it will actually be taken back to the source. It's a retrograde tracer. For some anterograde tracing, I think, for example, I can go the other way. I can inject into the VTA, and then I can see where the targets for the VTA are. So I'm not really sure what targets there are from the VTA to other structures. Injected into the VTA, gets into the VTA, and then it gets released at the level of the different targets. So, and this is an anterograde viral tracer, and you can use things like herpes simplex virus type 1 for anterograde, or retrograde pseudorabies virus. You can also have other markers like wheat germ agglutinin, and I'll come back to wheat germ agglutinin later, and that's a pure transneuronal tracer that we'll talk about, and the mechanism we'll talk about a little later. I bring all of this up because this is actually seminal to the Chowdhury paper. In the Chowdhury paper, the second half is actually more interesting than the first half. In the second half, and I'm pretty sure that they were asked to do this, in the second half, they took, remember, this is the original construct that they were using. They took this construct and they injected it into the VTA, and when it's in the VTA, every neuron within the VTA actually has Cre recombinase, and the Cre recombinase cuts at these sites, and because it cuts at these sites, and because they are non-symmetrical sites where they're facing each other, whatever sequence is in between them gets inverted and it becomes activated, which is why we see that channel rhodopsin being expressed. And that only occurs in the VTA. And the VTA, as I showed you in previous slides, using new N staining, definitely makes projections to the nucleus accumbens. But I just told you, and your colleague mentioned this as well, it also sends projections out to the prefrontal cortex, the habenula, as well as other areas. And one of the things that you have to know, because this involves stress, because this is a behavioral condition, where where are these targets and why is dopamine release all that important? Is it really dopamine affecting the nucleus accumbens, or could this actually be all the effects that you see where you're changing the firing rate in the VTA? Is it actually a change in the firing rate in the VTA, for example, to the medial prefrontal cortex? Maybe they just handle stress better. Maybe it has nothing to do with this reward system at all. And so one of the things that I'm sure that they were asked was, can you actually show that it's the projections that are going to the nucleus accumbens that are important? And so one of the things that was done in this paper, which is an incredibly difficult paper, was that they actually injected in, and this again is a really tough one, and I'll have to take you through the rationale for this. This is actually a really busy slide, although it doesn't look really busy. I just showed you this previous slide. PRV, what is PRV? Sorry? Pseudorabies virus retrograde tracer. So in other words, if you inject it in the target, where does it go? It goes back to its origin, the origin being the VTA, right? So imagine that you have this pseudorabies virus, which is a retrograde transneuronal virus. It gets taken up from the target and goes all the way back to the cell bodies that it started from. And you're injecting it in the nucleus accumbens, and you know that one of the potential targets is way out here in the VTA. So now let's not use those TH positive Cre recombinase containing animals. Let's not use those, okay? Let's not go back to this original condition that I showed you. And now let's start back at first principles. Let's say I take this construct now, which has the same Cre recombinase sites, same sites. But now I'm not using those mice that I showed you earlier on, the mice that had TH with Cre recombinase in them. I inject in this construct, but it's just a normal wild type mouse. If I'm doing nothing else to a normal wild type mouse, do I expect to see this reaction down here on the bottom? No. Why? Yes.
Because it doesn't contain the recombinants needed to splice the
Right, a normal mouse does not have this Cre recombinase. So how do I make sure that this is the pathway, the changes in the action potential firing between the nucleus accumbens and VTA is actually the cause for this? How can I actually show that the neurons that are going from here to here are actually the ones that are changing after optogenetics? That in fact, you know, they do send projections out here to the prefrontal cortex. How do I ensure that it's this pathway that's so important? I have to do this experiment again, which takes months at a time to be able to do, which is why it was originally submitted back in March, and it took until October for it to eventually become accepted. So what you have to do is take that pseudorabies virus, and now in that pseudorabies virus, include Cre recombinase. If you have pseudorabies virus that contains Cre recombinase, it still acts like pseudorabies virus. You're just bringing along with it Cre recombinase. And so what happens is, in these normal wild type mice, you inject into the nucleus accumbens pseudorabies virus that contains Cre recombinase that gets transported back to the cell bodies. If you are stereotactically injecting in the nucleus accumbens, you will not be introducing this Cre recombinase at the prefrontal cortex. This will definitely show you that any of the optogenetics that's being generated here at the level of the VTA, if this reaction does occur, where you're flipping it around and actually producing channel rhodopsin, if you put that in here and it's reacting and it actually forms channels, that you can actually have a specific pathway that you're looking at. Does that make sense for you? Yes? No? Sort of? So again, this goes back to the fact that if I use those original mice that this VTA has a number of projections, I'm only looking at one subcluster, only the TH positive neurons that are going to be making contact only with the nucleus accumbens are going to have this have the activated channel rhodopsin, because I'm providing at the target this Cre recombinase, and only the target will be able to generate this channel rhodopsin containing construct. Yeah.
So rather than putting it in the origin and then hoping that it's in the one pathway we're analyzing.
Yeah.
Put it in the target and then watch it go back to the origin and do its job again in the same
Yes.
pathway.
Yep, it's the same sorts of experiments, but now they're looking at just the pathway that connects the VTA to nucleus accumbens. It's very, very specific. It's a really tough experiment to do. So if you think about it, you're actually doing multiple injections, you're creating multiple constructs, you have to allow for expression of both, and then you have to be able to time this perfectly, which again is probably why it took several months or half a year for them to be able to do this. And they were able to, by doing those experiments, they were actually able to show 100% that yes, it's the VTA to nucleus accumbens, the specific pathway that is involved in this. So that was one aspect. Yeah, you have a question?
That does actually not say that the other pathways are not kind of could also be in the other ones as well.
Again, I'm not going to get, if you're really interested in it, you should go back and read the paper. It's a fairly interesting paper to read. I didn't show you all the results. And one of the things that you can do is inject into multiple areas, see if there are overlapping patterns of staining. And within the VTA to nucleus accumbens or medial prefrontal cortex, there don't seem to be any overlapping pathways that they were able to detect, or very few. So if you saw a connection between VTA and nucleus accumbens after this, it was pretty much because of that specific pathway. Do you have a question at the back? So sorry, why did they not use? When I say wild type mice, I mean mice that don't contain TH positive neurons that have Cre recombinase. They're still using those subthreshold mice that have chronic levels of stress that don't exhibit depression yet. They're able to reproduce everything in the same way, but what they showed was that it was a VTA to nucleus accumbens specific pathway that was changing that allowed them to fall into depression easily with phasic firing pattern changes. A lot of you look confused. Are you guys all okay, or can I go on? There's actually a couple more really cool experiments that I want to show you relating back to different types of vectors. So phasic firing of these VTA dopaminergic mice in those sub-threshold mice was really important. Could you actually convert mice from one phenotype to another? In other words, if it's only the firing that's really important, can you actually start going back? Once you've established the pattern, in other words, once I've stressed out a mouse and that mouse has continually had like tonic activation and it doesn't really change its firing pattern, it's been resilient so far, can I then go back? It's still had stress, by the way. It still had stress, but it hasn't changed its firing pattern. Could I go back and take those resilient mice that have been stressed out, haven't changed, and can I then go back and look at a change in the firing pattern? So what they decided to do was, rather than go back and try to use different constructs, they went back to their standard repeated 10-day social defeat model, not the one-day sub-threshold one-day stimulation model. They went back to the original type with the social defeat, and they identified different subgroups of mice. Some mice have depression, some mice are resilient to this social defeat after 10 days. And what they decided to do was, they were still stressed out. One group was resilient. It doesn't change its action potential firing rate. Can I then go back in and then see if I can change their firing rate optogenetically, and can they then fall into depression? And again, if you go back in just after 10 days of social defeat, and you look at a control mouse, this is the normal firing pattern in the VTA of a normal control mouse, no stress, nothing else has gone on. This is the normal firing pattern in the VTA neurons that you are able to isolate of a resilient mouse 10 days after it's been stressed out. And this is what happens if the mouse, after 10 days of social defeat stress, has fallen into depression. It increases its firing rate. Is that okay for everyone? This is the normal 10-day social defeat model. you can definitely see a change in the firing rate of action potentials. Can you all see that if you are depressed or you're susceptible to depression, you increase the number of action potentials over the same unit of time? If you are not going to be depressed, your firing rate stays the same as a control mouse. You all see that as well, right? Okay, perfect. Again, they show in a normal mouse, nothing is done to them. The firing rate in a susceptible mouse, repeating the experiments that were done by Eric Nestler's group many, many years ago, shows exactly the same thing. The firing rate definitely goes up. Now, if you do the optogenetic experiment, you introduce channel rhodopsin into the VTA, and then you do phasic stimulation or you do tonic stimulation of these mice. Remember, they have been stressed out, okay? Even the resilient mouse has been stressed out. It's just been resilient so far and hasn't changed its rate. So if I go back in and I take a look at these resilient mice, do these resilient mice change their firing pattern after all of this has occurred? And yes, they do. You can actually convert a resilient mouse by having phasic firing into a depressed looking mouse, okay? And the social interaction is definitely decreased, as well as the sucrose preference. Anhedonia is observable, as well as a decreased social interaction, both signs that we've looked at for depression in the social defeat model. So different conditions, a resilient mouse who has been stressed out can actually fall into a depression-like state by having this optogenetic stimulation. You can actually go in and do the reverse situation as well. I'm there, thank you. Okay, good. Yes, Jessica. Sorry? Yeah, the sucrose preference index goes down in following phasic stimulation in blue. Yeah, the color scheme up here doesn't match the color scheme that's down here. It's a different sort of set of experiments. Okay. It's one group of mice. It's all resilient mice that you now introduce channel rhodopsin or the control EYFP, and then you stimulate phasically or tonically on the bottom two panels. Okay. So can you go back in and do the reverse? I told you that you have two different ways in which to modulate the activity of neurons. You can use channel rhodopsins, which excite, and you can also use halorhodopsins, which actually do the reverse and actually cause inhibition. And if you go in and use one of the versions of halorhodopsin, which is HR, a particular version of halorhodopsin called version 3.0, I think they're up to like version 4 point something now. One of the things that you can see is that following stimulation, not by blue light in this case, but by yellow-orange light, that you can actually cause an inhibitory signal to be generated. So here's the normal spiking pattern of the VTA neurons, and in the presence of this halorhodopsin, when you turn on the yellow light, it stops its activity altogether. Only when the yellow light is on. As soon as you turn the yellow light off, it goes back to its regular firing pattern. Is that okay for everyone? I think that's fairly easy. So again, using the same system that we talked about before, only at the level of the nucleus accumbens, injecting in that pseudo-rabies virus containing Cre recombinase that gets retrogradely transported back, and now injecting in the adenovirus that contains halorhodopsins, they're able to show that you can reverse the phenotype if you have a mouse that is stressed out and that you pushed over the edge by increasing its phasic firing rate, and now you take and put in this channel rhodopsin using that social defeat model that I showed you, 10 days of social defeat, increase the firing rate. Now I go in and introduce halorhodopsin. If I block that increased firing rate, do they behave normally? And exactly as you predict, they do. So it was a remarkable series of experiments in back-to-back papers that showed that changes in the firing rate between the VTA and nucleus accumbens. And again, hopefully now you'll understand why we say it's VTA to nucleus accumbens, it's not other areas, is specific enough to elicit behaviors that lead to either depression or to resilience. Yes?
For both these examples,
Yeah.
whether it be making resilient mice susceptible or making susceptible mice resilient,
Yeah.
this is after they have went through their 30
You-
days and they've already settled in as being either resilient or
That's correct.
Can you use, because what they did in the original experiment was they altered their firing while they were stressing out the mouse, right? While the phasic firing.
Not in all situations.
Okay.
In some models, they were already entrained in one pattern or another. They already had social defeat. This, for example, they already had this entrained, and then they went back and did this experiment with channel rhodopsin. Same thing here. They're already resilient or susceptible, and by inhibiting them, you can actually show that you can reverse the susceptible mice back to a normal phenotype.
But my question was going to be, can you, while you're originally, while you're, I guess, originally going through the social defeat model,
Yeah.
can you create resilient mice by stimulating halorhodopsin while they're getting stressed out so that they don't ever...
So that's an interesting model. So if you prevent this from occurring in the first place, my prediction would be that you probably could, but they didn't do that in these experiments. Yeah. Yes.
Would you be able to discern from these experiments whether the synaptic input is heterosynaptic or homosynaptic?
You wouldn't be able to. Not in these experiments. Any? Yeah. Do you have a question?
Yeah, just the events. So they did 10-day social defeat.
In some models, they did 10 days of social defeat. In other ones, they did sub-threshold or suboptimal stress levels where they didn't exhibit depression. It depends on which experiments you're looking at. They have a number of them.
Okay, but here, like, they
Yep.
got the depressed mice, like they
Yep.
got the mice who were depressed.
Yep.
Then they put in the construct, the halorhodopsin construct, and then they saw this change, but they weren't depressed anymore.
Yep. That's correct. Yep. Okay, so you can change the overall firing, which is basically what is shown here. They increase their sucrose preference after halorhodopsin. After they've already been susceptible, they go to being resilient. Don't have to worry about the changes in magnitude or anything else. It's just to show you that you can reverse the phenotype. You can make a resilient mouse susceptible. You can make a susceptible mouse resilient simply by changing the firing pattern between the two different segments. They went even further, and again, I won't go through all of the different results that are shown to you on the left. If you really want to go through it, you don't need to worry about it for your test, etc. If you really want to go through it, we can. They looked at the same thing in the medial prefrontal cortex and VTA group of neurons, and what they showed was that there aren't any effects that are being mediated between the VTA and medial prefrontal cortex. So they're ruling out that any of the changes that they're seeing induced in these models are actually involving these VTA to medial prefrontal cortical dopaminergic neurons. Okay, they don't see the same effects. They can't reverse things if they put in channel rhodopsin and they're looking at changes in the firing rate between the two. They really don't see the same types of effects that they saw with the VTA and nucleus accumbens. If you don't mind, I'm going to go through one last two slides and then we'll take a break. I know I've gone a little bit longer than I normally do. So there's a lot of relevance for us, and there's a lot of really important cellular molecular biology in the experiments that I've just shown you. I lied to you earlier on. I lied to you in the sense that I told you that if you are on the antidepressant medication, that it takes a long time for that antidepressant medication to work. Do you remember that lie? That it takes 3 to 4 weeks, and that one of the reasons why we think it takes 3 to 4 weeks is what's known as the neurotrophic and neurogenesis hypothesis. It takes time for neurons in the dentate gyrus to be born and to incorporate in the hippocampus, and that's why antidepressants take time. However, we know, for example, that in certain antidepressants, you can actually see an effect within several hours. The mechanism for that has never really been explained. You can, for example, have treatment-resistant depression, go in and get a shot of ketamine, and ketamine will reverse the effects of depression in some individuals almost immediately. So I'll get into that in a moment, but these rapid antidepressant effects, like ketamine and others, might be important because this might explain it. If I can change your phenotype within a matter of a few hours by activating channel rhodopsin and changing your firing rate, maybe there is a possibility of being able to do this. This IH current seems, or this hyperpolarization activated current is really key to this whole process. Why do we see an increase in the firing rate between the VTA and nucleus accumbens? Why do we see this transition from tonic to phasic? It's believed to be this IH current that allows this switch and transition to occur, and it might involve things like HCN2, which is a channel protein that's believed to mediate this IH current. This HCN2, by the way, is highly expressed in the VTA to nucleus accumbens pathway. And so it makes sense that you're able to switch, turn on or turn off this IH current relatively quickly. This IH current is also really turned on in susceptible mice, and IH inhibitors display this really rapid, long-lasting antidepressant effect, which might actually tie everything together. That paper has not come out yet. There are a lot of rumors to suggest that the rapid antidepressants are being examined in these models, and that someone will be publishing that really soon. And this gets back into these rapid acting. Ketamine is one way that you can rapidly reverse depression. So some individuals, different fluoxetine, for example, it doesn't work for you, it takes several weeks, doesn't work for you. You go in, you get a shot of ketamine, and what people describe it is like, wow, suddenly everything looks brighter. I feel like I'm like out in the sunshine, everything feels great, ketamine. By the way, this is one of those paradoxical things, and if you're interested in doing this, you should follow up in one of your fourth-year sleep classes. Sleep deprivation is one of the triggers for depression. Prolonged sleep deprivation has also been shown to rapidly reverse the effects in some individuals of depression. So you take a depressed individual, just don't let them sleep, and they will get better. Again, not really clear as to why. Deep brain stimulation, you target certain areas of the brain. We talked about area CG25. You turn on area or inhibit area CG25, and they get better right away. So we know depression can be turned on and off really, really quickly by a variety of chemicals, stimulation protocols, or physiological mechanisms, but no one has been able to show that. This paper demonstrates that at least using molecular cellular biology that you might be able to do this as well. So this might get into this new idea of these rescue effects in this particular pathway. It's still really early though. Not entirely sure if this pathway is going to be of therapeutic benefit, and that remains to be seen. I mentioned at the very outset that now with different forms of optogenetically activatable G protein coupled receptors, it's theoretically possible to do this. But however, I'm going to take you through the classic technique that's available right now for G protein coupled modulation. And this gets into what's known as DREADS. And this DREAD technique is designer receptor exclusively activated by a designer drug. You'll understand why we call it DREADS in a few moments. So this will control whatever type of neuron that you can introduce this construct to. You still do it the same way. You have a construct that you generate. After you generate the construct, you need to use a virus. It infects these neurons. And then instead of using light, you now will start using drugs to be able to do all of this. So this will incorporate a synthetic receptor. It's normally not found in nature. It doesn't exist within you. It doesn't exist within different animals. It's a totally synthetic receptor that we've designed. It, again, one of the ones that I'll show you is this HMX-DX family, and you can incorporate that by viral expression into different cells in the animal's genomes. You have to decide which cells you want to target. Same ideas that we talked about before. You need the machinery. It's a viral infection, and you need to be able to express this transgene. Same ideas. You need to know what virus, speed of delivery, speed of expression, length of time of expression, etc. Exactly the same things, but DREADS are just a little bit different in terms of how you turn them on. So these are G protein coupled receptors that have been genetically engineered. There is no endogenous ligand. They do not normally interact. If you express this in the brain of an animal, it doesn't work at all. It's just like it's not even functionally there. There's no baseline activity. There's no change in any type of activity in any neuron until you provide an exogenous synthetic ligand. So the construct, the receptor is synthetic, and so is the drug. The drug itself doesn't even look and mimic like a neurotransmitter that you would normally have. And this ligand is called CNO, and you'll often see it abbreviated as CNO, and this is clozapine N oxide. And again, it's something that you inject into the animal to activate these receptors if they get expressed. So you can see that in the hippocampus, for example, you get good expression in red of this particular G protein coupled receptor, and then you can see the activation takes a little bit of time. It's longer in terms of activation than you would have with optogenetics. Optogenetics, you turn on a light, the channel opens like right away. Here, you inject a drug, it might take minutes to several minutes for you to actually see activation of these G protein coupled receptors. There are a number of different flavors that are available for activation. So just like with optogenetics, we have halorhodopsins and we have channel rhodopsins, inhibitory versus excitatory. CNO can interact, and normally it was derived from this muscarinic receptor. The muscarinic receptor normally has acetylcholine activating it. It's been genetically engineered to no longer interact with acetylcholine, and it only interacts with CNO. And that's the only thing that it would interact with. No other endogenous ligand or neurotransmitter will turn this on. And depending on which version of this you have, whether it is M3 or whether it is M4, it will activate different G protein coupled receptors. It might, for example, couple with the GQ11 types of G proteins, or it might interact with GI or GO types of proteins, which cause neuronal silencing. In other words, you are able to turn on neurons, excitation with HM3D, or you can turn off neurons by introducing HM4D. Depends on what you're trying to do. Again, there are a number of different variants. You don't have to worry about all of them. I'll ask you to know the human variants that are often used in animal studies rather than the rat versions of these, which is why you have R versus H, but all really important in terms of specificity as well as a specific ligand. They will not get turned on until you inject CNO, and you determine when you turn these on by injecting in CNO. Do you have any questions about this before we go on? Yeah.
Are both, like the M3 or M4, are they both equally susceptible or equally responsive to CNO?
Yes.
If you were to put CNO in both receptors, like both receptors are present, and you put in CNO.
That would be a really bad experiment to do. Yes. Yeah, you would have to do a couple of experiments where you introduced this into one subset of neurons, and then you'd have to see what the expression levels are. But yeah, in theory, they're equal. Any other questions before we go on? Okay, and again, don't worry about the fact that this is a really busy slide. It's just to demonstrate a couple of things. You can actually inject in the virus into different areas of the brain. In this case, one of the things I'm sort of interested in is feeding behavior and other types of behaviors in groups of neurons found within the hypothalamus. And you can see that you get good expression in a group of neurons. You can go back and track the expression of neurons in these different colors, and that you can see that when you add CNO, it takes a little bit of time for this to actually occur. The time frame down here is not on the order of milliseconds, it's on the order of minutes. And so when you inject in CNO here, it takes several minutes for CNO to actually cause a change in activation, and it takes an equally long period of time to actually see changes in behavior, which is what these different graphs are showing you. This is really just to illustrate, and it's not something that you should memorize, that both the receptor activation and behavior using DREADS is on a different time scale than you would see with optogenetics. Okay, and that's all you should be taking away from this particular slide. And again, you can do the same types of experiments that we did with optogenetics. You would have to inject in different viruses, and you'd have to design different DREADS accordingly. When you start looking at the comparison between DREADS and optogenetics, both techniques are specific to the specific cell types that you want to be able to target, as well as the groups of neurons in specific brain regions. I could use, for example, DREADS in my VTA nucleus accumbens experiments if I really, really wanted to. You think it would be a good idea to do that in the experiments that I showed you? Would I be able to show a difference between tonic and phasic activation using DREADS? Probably not. And so there are reasons when, or times when I want to use DREADS, when I want to use optogenetics. And I'll go through some of those a little bit in this particular slide and on the next slide. So again, simple differences. DREADS are activated by CNO versus light activation of opsins. DREADS have a relatively slow minute time scale versus the channel rhodopsins, halo rhodopsins, acting really rapidly on the order of milliseconds. Generally speaking, right now the move is away from DREADS. Richard Roth's original sort of use of DREADS has now been usurped by the use of optogenetics. There are some exceptions. When I need to do prolonged behavioral testing, which is why I showed you that previous slide, the arcuate nucleus that's involved in feeding behaviors in the hypothalamus and other behaviors in the hypothalamus, if I'm looking on a long-term time scale, and I have to be able to see this on a long-term time scale, days to weeks, there's really no preference between having optogenetics versus CNO. The one big advantage here is in CNO, all I have to do is inject in systemically, like IP or through a tail vein or something else. CNO, it gets into the bloodstream, it gets into the brain. And so it's a simpler process than having to put in a fiber optic light source through the skull. Do you have any questions before we go on to the last technique that I want you to really be comfortable with? Yes, at the back. Yeah. No, they don't normally exist in the brain, so no, they wouldn't. Yes. Yeah. Well, that's a very almost philosophical question, to be quite honest. Like, if you have a cell, how many different receptors can you put on a cell without affecting the cell? Or how full can you get, depending on how much food I give you, right? I'm not really sure, to be honest. There might be a certain number or set number of places that a receptor can be expressed on the cell surface. It has a finite surface area, so I'm not really sure. It could be altering function. You're absolutely right, but I don't think that in normal circumstances that ever gets into or taken into serious consideration. Okay, I'm going to go take you through this last paper, and this last paper is actually a really important paper. We have met many of these individuals several times in this course. So we have already talked about AJ Silva. Do you remember AJ Silva at all? Where have we seen AJ Silva? Oh, everyone's memory being jogged right now. Go from a jog to a really rapid sprint. Can you do that? AJ Silva, do you guys remember the original experiments that involved CAMK2 alpha? In the CAMK2 alpha, there was a change in phosphorylation. This is that AJ Silva. So AJ Silva was also really important because two individuals that are really famous here in Toronto, Sheena Jocelyn as well as Paul Franklin, they did their postdocs with him down in the United States, and they published a series of papers based on all of the ideas that they developed down in UCLA with AJ Silva. One of the things that seems to be really important is that different groups of neurons. So I mentioned this really briefly in passing. The different groups of neurons seem to be able to almost fight for memory. And this competition for memory is something that we talked about in neurogenesis. In the neurogenesis, we talked about why you can't remember things from before you were 3 years old. And it's thought that the newly born neurons have stronger, more pliable synapses that are stripping away and competing out for synapses that are already there. And this is why you can't remember things, because no memories are actually stored because they've been competed out. this gets into this other idea that neurons that you normally have, why doesn't every last neuron have the same capacity to be able to get involved in memory formation? And this is one of the things that Sheena Jocelyn with AJ Silva was really interested in. And I'm going to take you through some of the results from their paper, and along the way start introducing some of the concepts of that paper to you as well. I'll ask you to remember for the time being John Guzowski, as I'm going to introduce one more technique to you. We've talked about this model several times, right? Can anyone walk me through this model? Like, what are we seeing here in this particular model? So we're seeing conditioning to what? Sound, okay. And shock, okay. So what happens is, normally we allow the mice to habituate. And what is habituation? Chill out, okay, nice. How about not chill out? What else could we call habituation? Yeah. Ignore a non-threatening stimulus. Just getting used to the surroundings, right? You've all habituated to being in this classroom. It's fairly non-threatening for you. You've never written a midterm here. Unlike EX, you know, you probably find EX really threatening, and you probably never want to go over there. So habituation is just getting acclimatized to the box, and you see that the animal doesn't freeze at all, no matter the length of time. It just freely goes around and checks out what's going on there. In day 2, you can do some conditioning. And so what happens in conditioning, in this conditioned stimulus, is that on the first day that you go in, you pair, you give some sort of sound, and then as this sound is playing, at some point during the sound being played, this conditioning stimulus that you see here, maybe between 20 and 30 seconds of music playing, in the last part of that music being played, one of the things that you do is you provide a little foot shock to the animal. And as you provide a little foot shock, the animal stops moving. It freezes. And as it learns that this music precedes the delivery of a foot shock that it doesn't like, it starts freezing more and more often on every trial that you give it. The more often it hears this music, the more often it will start freezing, and you can start measuring this behavior. And so over time, this animal, even without foot shock, will have this type of behavior where it freezes all the time, only in response to this auditory stimulus, this music that's being played, even without the foot shock being delivered to it. So this is one of the things that you need to keep in context as we go through this next set of experiments. So a lot of work, just like we looked at depression, a lot of work had shown that the lateral amygdala has increased levels of CREB in that model that I just showed you. As it's learning that sound precedes a foot shock, the levels of CREB in this area of the brain known as the lateral amygdala increase. And a lot of different groups had actually shown this. If you go in and you have neurons, you can actually see that there are neurons that overexpress CREB. There's lots and lots of CREB found in these particular areas. You could also do an experiment where instead of just looking in the lateral amygdala and seeing the levels of expression, you yourself can go in and inject in a virus that overexpresses CREB. And when you do this, that they're more likely to be activated, and they're more likely to learn this faster and remember it longer if you overexpress CREB. So CREB had been hypothesized for a very, very long time to be the key molecule that's involved in this contextual learning, the association between sound and this auditory learning that is found in the amygdala, the lateral amygdala. Is that okay for everyone? So how can we go about proving that CREB in this area of the brain that's been associated with learning, this fear learning, this auditory fear learning, how can we go in and start lesioning these neurons? The same idea. Remember, with electrical stimulation, with lesion studies, you have to be precise. You can't be like scooping out whole areas of the brain and saying, I scooped this area out, therefore the lateral amygdala is involved. How do I only lesion the lateral amygdala, and only lesion the lateral amygdala that contains CREB after this learning has occurred? That's a really, really tough proposition. So again, just as a review for you, CREB was one of those molecules that we looked at earlier on. So we know that within the lateral amygdala, the same types of things that we saw with learning and memory in the hippocampus also occurs in the lateral amygdala. So you'd have to imagine that this is the neuron, the postsynaptic side of a neuron in the lateral amygdala. We know that this process too, just like with learning and memory in the hippocampus, is also protein synthesis dependent. You can block Krebs expression, you can block the freezing. They won't learn to freeze with music by injecting in cyclohexamide or actinomycin D into the basolateral amygdala. They will not freeze. We also know that this form of learning involves GLUA1 or GLUR1, so it's an AMPA receptor mediated plasticity. And there also seems to be downstream molecules that we've seen before that have been shown by a number of different groups that seem to be activated in this particular model. So as the animal learns that music is something bad, I should freeze because I might get a foot shock, in this particular neuron, you might start to see the increased production of BDNF. You might start to see ARC being expressed, which is an immediate early gene. You might see cytoskeletal proteins being overexpressed, ZIF268, Homer 1A, and there has been some implications that in the lateral amygdala, PKM zeta plays a key role in this as well. That hasn't been discredited so far in this area of the brain. We also know that it's RNA synthesis dependent, just like we saw with the long-term memory formation that we saw elsewhere. So many of the same systems that we saw before. So how do we go in and measure activation? And this is a technique that I'd like you to be familiar with. It's a relatively new technique. It's known as catfish. And catfish is a technique that as science students, you should be familiar with. You'll read these in a number of different papers. So catfish stands for the cellular compartment analysis of temporal activity by fluorescence in situ hybridization. The fish part is this fluorescence in situ hybridization. So here are a group of neurons, and you can see the nuclei are stained in blue, probably through DAPI staining that we've looked at elsewhere. And one of the things that you can see on that previous slide is that as an animal learns, in this learning process, there are a number of these immediate early genes that get turned on. Fluorescence in situ hybridization detects RNAs, okay? It does not detect protein, is not an antibody-mediated staining procedure. It is the complementary RNA being bound to the endogenous RNA that's being expressed. So this catfish method actually detects the activity of different neurons being turned on. It will tell you when neurons are being turned on, and it'll tell you which neurons are being turned on. So for example, in this particular case, this one molecule that we're measuring, this is ARC. As I mentioned to you before, ARC or ARG 3.1, it's the same immediate early gene. When this neuron becomes activated in the lateral amygdala, you could look, for example, in the hippocampus. When that neuron gets turned on, the levels of ARC go up. Do you see, and ARC is here in red, okay, and or pink, depending on how you're seeing it. Do you see a lot of ARC under this condition? Is there a lot of ARC fluorescence? So there isn't very much ARC fluorescence here at all. After the animal has started to learn this procedure, the first 5 minutes or so after the first foot shock, if you were to take the animal and you were to take out the lateral amygdala and you were to do the staining 5 minutes after that shock, what you would see is that ARC RNA starts to increase. If you let that animal, instead of 5 minutes after it had just precedes a foot shock, if you were to then take that animal and let it go for about half an hour to an hour and then examine the same area of the brain, you'll see a couple of things. What do you observe down here in C? Yes? So what was the center? So what is this blue thing staining? No, it's not a synapse. What's this blue stain here? So this is the nucleus. Here's another nucleus. Here's the third nucleus, 4, 5. All of these are nuclei, and you can see that ARC is in the nucleus here, right? 5 minutes after it's learned, nuclear expression of ARC. Where do you see ARC after about half an hour to an hour? Outside of the nucleus. Anything else that you observe about this? Put on your scientific hats. What else do you see? Could be traveling, it could be moving, and remember RNA gets captured at synapses. Do you see synapses here? Probably can't see synapses here. What else do you see about ARC? Sorry? Is it breaking down? No, it shouldn't be breaking down the nucleus. Sorry, there's a hole. It's probably the nucleolus. The nucleolus is full of proteins, doesn't stain with DAPI. Anything else? Yes. Your colleague at the front mentioned the same thing. Perhaps. Anything else? Just like a regular observation. Yes.
It's free floating. There's some red that's not at all attached to anything.
Maybe. Where might it be though? Or it might be in the cytoplasm, right? So, or in the cell body. Anything else? Like just any observation. So specific neurons, sure. We saw specific neurons up here, but what's the difference between here and here? There's a lot more. That's right. You see a lot more ARC. And so there's a slow buildup and there's a slow translocation of ARC. And that's been known for a while. ARC is an activity marker. If I'm looking at these neurons, these are the neurons that are learning. Do you see it in every neuron? No. Only some neurons have activity and only some neurons are learning. Not all neurons are learning. Why is it that some neurons are learning? And how do we know that these are the important neurons in learning? And that's the fundamental question that's being asked here. Okay, so I'm going to take you through, this is the area of the brain that we're looking at in rats. This is the lateral amygdala. And the lateral amygdala here, either through staining for all nuclei or staining specifically after viral infection, this is the area that we're looking at. You can see that the viral infection here is pretty good. It's targeted and it has hit many of the neurons within the lateral amygdala, but you'll see a few that are outside of the lateral amygdala as well. And one of the things that had been known is that if you follow ARC and then you go back and do staining for CREB, the ones that showed activity, ARC activity, are the same ones that overexpress CREB. In fact, we look at a certain form of CREB, which is known as serine 133 CREB. CREB is sort of the master learning molecule, we think, right now. So PKM zeta was the old molecule that everyone was looking at. The new one that everyone is looking at is CREB. And we can tell if CREB is being turned on. If CREB is activated in learning, then we will see more phosphorylation of serine 133, which is why we see an increase in phospho-CREB, phosphorylating serine 133 after tone and shock. This animal is learning. If we go in and we stain for serine phosphoserine 133. We know that CREB is important. The more phospho-CREB that we have, the better the freezing. If you have a CREB knockout animal where there's no CREB in the lateral amygdala, no freezing will occur. They won't learn this easy task. So we know that CREB is really important. And again, all that's being shown down here is that without CREB, you really don't have learning. If you reintroduce CREB, you can actually introduce learning. So a knockout animal, a CREB minus animal that normally doesn't learn, it would look like this. If you go back in and now you express CREB in these neurons, it will go back to that same freezing behavior that we saw before. So hopefully you'll buy into the fact that CREB is really important in this process. We can measure it in a number of ways and that we can see activity dependent on ARC. Yes, at the back. Yeah, that's a great question. To be honest, I'm not sure about phosphatases. They have used other kinases to push this process a little further. I'm not sure about phosphatases. So again, a busy slide. You do not have to memorize any of the slide here on the left. It's really what I want to show you here. If you go back and you look at the difference between ARC and CREB, wherever you see ARC, activation, activity dependence, you will also see CREB. In other words, the neurons that are getting turned on that we can measure through ARC activity, through catfish activity, and we go back and stain for CREB in green, they correlate very, very tightly. So in other words, CREB is found in neurons that are activated in learning. Is that okay for everyone? So ARC positive, CREB positive activity, as well as a learning molecule. And we think that this is why we have certain neurons that are important in certain forms of learning. Again, just a really quick review of things. We know that activated synapses will signal through CAM kinase 2. It could eventually lead to activation of MAP kinases. MAP kinases in turn can translocate to the nucleus. That was that busy looking slide that I showed you with the AMPA receptors, and that this MAP kinase translocation can cause the increased phosphorylation on serine 133 of Krebs. And it's the phospho-Krebs, and you'll have to remember this for Dr. Eubanks when he comes in and talks to you about epigenetics. It's phospho-Krebs that is the true transcription factor. This phospho-Krebs is the one that binds to nuclear sequences and that will turn on those DNA, the different genes that are associated with these DNA sequences. This phospho-Krebs can also bind to Krebs binding protein, which you'll also hear from Dr. Eubanks. Both of these, as you can see, it's a central player. You'll see that if you are missing CBP or you're missing phospho-Krebs, you're going to mess up synaptic development, and he'll show you that from an epigenetic standpoint. You need this pathway in order to increase other factors like CFOS, ZIF268, BDNF, etc. So we've seen that contextual fear, auditory conditioning, all of those things evoke a fear response that we can pick up in the lateral amygdala. It looks a lot like LTP, involves glutamate receptors, and involves CAM kinase, translocation of MAP kinase. And we can also make use, just like Carl Diceroth did earlier on, of a unique mouse that was available to everyone. And this unique mouse expresses what's known as diphtherotoxin receptor. And it's an inducible diphtherotoxin receptor. And I'll get into why that's important in a moment. And it has this, by the way, in every single cell in its body. It's not unique to the brain. This receptor is normally not expressed in mice. In a lab setting, the mouse are diphtherotoxin, so it's never, sorry? Dip, H-E-R-I. Yeah, it takes a little while for the old brain to get working. So what you can do is you can go in and create a specific vector that incorporates things like Krebs with Cre. So we've seen a rabies virus or pseudo-rabies virus that incorporated Cre, and now we can actually attach Krebs with Cre recombinase. Keep this in mind as we start thinking about how you can use Cre recombinase in a number of different situations. And think about why we might need Cre recombinase in a few slides, or in this slide, never mind. Okay, I just told you that this mouse actually has this diphtherotoxin receptor. Okay, this diphtherotoxin receptor is under the control in every single cell that the mouse has. This is also in the literature also called the Rosa mouse or the IDTR mouse. It's all related to the same thing. But take a look at this, okay? And at this point, I'm actually going to ask you to tell me some information about this particular construct. In this mouse here, in the normal situation, the diphtherotoxin is downstream here. And upstream of this, we have this stop codon. Going back to your first year or maybe even high school, will diphtherotoxin ever be expressed? There's a stop codon preceding it, and so that doesn't ever occur. Now, also take a look at this, and take a look at this in the context of LOX-P. I just told you, or we had several lectures now, 2 lectures now, about Cre recombinase and LOX-P. Look at the orientation of LOX-P. What will happen if I introduce Cre recombinase to this particular construct that I have here, where the LOX-P sites are flanking this stop codon? Right, so remember, these are now in the same orientation. They're not in opposite orientations. Opposite orientations means that whatever's found between them gets inverted or flipped around. If they're in the same orientation as you see here, the arrows, the little triangles in front of and behind the stop codon actually show that they're going to remove this stop codon in the presence of Cre recombinase. If the stop codon is removed, then you will express diphtherotoxin receptor. Okay? Yes? Good. Is that a problem for the mouse? Yes? Does it normally ever encounter diphtheria toxin? No. So the receptor is there, but in a normal controlled lab setting, diphtheria toxin is never going to be something that the mouse encounters. But you can introduce diphtheria toxin to the mouse through systemic delivery. You can intraperitoneally inject in diphtheria toxin. It gets into every single cell, including cells in the brain. So why is that important? Any cell, which in this case is every cell, expresses this diphtheria toxin receptor. And when diphtheria toxin binds to its receptor, any cell that expresses that receptor will undergo apoptotic cell death. Those cells will die off, okay? And that's why we actually want to make use of this. So these mice have to be made to express this diphtheria toxin receptor. So is it possible to stereotactically, and again, that's one of the things that we talked about, stereotactic injection into an area of the brain gives you some control. Can we inject a vector that targets the lateral amygdala neurons and activates the diphtheria toxin receptor, but only the ones that are expressing CREB? Well, I told you on that previous slide that one of the things that we want to do is introduce CREB and at the same time introduce Cre recombinase. So hopefully now you'll start to be able to put together some of these different pieces. So what happened in this particular paper that I showed you earlier on, they created this construct that contains CREB, which will be injected into the lateral amygdala. And in the lateral amygdala, when neurons are learning that a tone precedes a shock, that in the lateral amygdala, CREB levels go up, that animal learns, and ARC goes up, everything is great in that particular group of neurons in the lateral amygdala. In this mouse, which can also learn this auditory association, the Rosa mouse has no problems learning association. This Rosa mouse has these LOX-P in the same direction, same orientation, LOX-P sites around the stop codon immediately preceding diphtheria toxin. I have stereotactically injected CREB and Cre here. What will this vector do if it gets expressed? Yes.
It will provide it with CREB and at the same time activate the LOX-P and cut away the stop codon. So now having CREB and
Perfect. So any cell that the virus gets into will first express CREB, or any neuron that it gets into will first express CREB, and it will also express Cre recombinase. And in those neurons that now express CREB and express Cre recombinase, you will now be expressing the diphtheria toxin receptor. Is that okay for everyone? So you've got overexpression of CREB. You've now activated a system that will activate diphtheria toxin receptor. So you've created this system where these neurons are actually going to start producing lots and lots of CREB and Cre in a group of neurons. Then you get these animals to start learning this association. And one of the things that had been known for a while, neurons that overexpress CREB, they're the ones that are most likely to learn. If I really wanted you to remember and learn everything, I would just fill your brains with nothing but CREB all the time. In fact, I do something similar. Instead of filling it with CREB, I tend to fill it with something that sounds like CREB. Yes, we won't go there. I'm glad that you were able to pick up on that fast. So when you start overexpressing CREB and Cre, these are the ones that are actually going to be picking up that fear memory. Other ones will be as well, but these are the ones that are more likely to be able to pick up that learning, that memory that's going up with fear conditioning. So all of these cells are going to be involved. And these are the cells that are also expressing Cre recombinase. Does this cell express Cre recombinase? No, it does not. And so what I can do is I can go in now, I can inject in diphtheria toxin receptor, diphtheria toxin, and when diphtheria toxin goes in, have I expressed diphtheria toxin everywhere in this mouse?
Aren't you injecting diphtheria into
I'm injecting it systemically. It's going around. Where is the receptor being expressed?
the receptors
Only where there's CREB and Cre, and only in the lateral amygdala. And so when I go in, these are the ones that learned. All three of these learned the fear association. Now I have specifically and very, very specifically only removed the ones that were involved in the fear learning. I have not damaged any of these other lateral amygdalar neurons. Do you all follow that? It's really important. Okay, so these other ones that might have been involved in learning might still be there, and these other ones that never got involved in learning nor took up the CREB or CRE are not going to be affected at all. Only these two will be dead because they express CRE, and now we've given it diphtheria-toxin. Now, I go back in. This animal had learned this task. What happens to this animal now when the memory has been wiped out of its lateral amygdala? What do you think will happen? Yes? Yeah, and again, that's exactly right. Unfortunately, I'm running a little bit short of time, so I won't go through this slide in detail, nor do you have to memorize the magnitude. But your colleague is exactly right. They freeze less. It's like they never learned that task. They had learned it originally. That memory was encoded in certain neurons in the lateral amygdala, and those lateral amygdala neurons that had the overexpression of CREB are now gone. They have been eliminated from this memory trace. And when you go back after diphtheria toxin injection, in fact, they freeze a lot less. It's like they never learned the association. So here, freezing more, the higher the bar is, the better off your learning is. And here with diphtheria toxin overexpression, you can see that the animal has not learned after you have killed off those neurons. So again, it was a really elegant experiment that I want you to be able to think about. How might you use that in different conditions? Me? Yes, they're fused. They're in-frame constructs. Yep, Jessica.
You know how you induce the CREB expression by injecting it? How are you? But we want to target only CREB expressing neurons, but like we're providing the CREB.
You are overexpressing CREB. Every neuron has CREB. You are overexpressing CREB. So in some ways, it's a lot like this. So if I gave everyone here like $100, right? And I gave everyone in this class about $100, or I gave some of you $100, and I asked you to go out and buy as much coffee as you possibly could. You could all go out and do that. Some of you will be able to go out and buy coffee even without me giving you $100, right? So you'll still be able to do that. However, if I then took that group of students away and then saw how many students were drinking coffee, I would only see the numbers that were able to buy it by themselves. Okay, and that's what you're doing. You're taking the group that you've over-advantaged that will learn that memory, and now you're taking those out of the equation. They learn better. The more CREB you have, the better you learn. You're more likely to pick up something than if you have basal levels of CREB. Okay, so any questions before I go on? Yeah, it's a very cool experiment. It's still considered a classic experiment. So this Rosa mouse actually allows this specific ablation, very, very specific ablation of neurons that overexpress CREB that have learned this fear response. The effects were also very, very specific, and this gives you another way in which to manipulate discrete groups of neurons and a way to eliminate or ablate specific groups of neurons that are associated with a particular memory. It is still to this day cited as a classic paper, and variations of this paper are still being published today as well. If you want to learn more about this technique, this is the paper that I just presented to you. This is that classic paper that was published in 2007. There are a number of follow-up papers, and some elements of this are also found in the 2009 paper that was published in Science. And you can see that these this sort of consortium of individuals. Do you remember who John Guzowski is? Who is John Guzowski? Oh, he did the ARC, the catfish. He developed that, and you can see why they might have him on this paper. And of course, Paul Franklin, Sheena Jocelyn, and a number of others were involved in this paper that I showed you about the selective erasures. So these classic papers still exist to this day, and they give us an idea of the specificity at which CREB works on different systems. So I have like 2 minutes before I have to let you go. So on your final assignment, what you should be trying to do, and it's due next Friday, what you should be trying to do is to have 1 or 2 sentences that summarize the overall results that were observed in the original paper that you did your summary on. So that should be at the very front. The second thing that you should try to do is find other papers that might fill in some of the gaps, the future directions that you're responsible for on this upcoming assignment. And so I would suggest finding about 2 or 3 other papers, maybe in a related field, where you think that the authors might want to follow up with like 2 different experiments. Most importantly, you should be able to give a rationale why you think those experiments would be important. And that's really what you're being evaluated on. Not just coming up with any old random experiment. I think they should go in and start staining for CFOS. Why should they stain for CFOS in this experiment? If you don't give me a good rationale based on other papers, you're not going to get a good mark. Okay? Do you have any questions before we wrap things up for the day? Yes, Ashkin. That's a great question. Ashkan, eventually you'll be doing sort of a critique of things that might not have been done well, or things that you would change if you were to do these same experiments again. But for this assignment, what you're doing is providing sort of a future direction based on the results that they got in this paper and based on this larger literature. These are the most logical experiments because of this rationale. And I wanted to see the rationale, I want to see that you've read other papers, and I wanted to see what your future directions are. And you guys will all do well, right? Good, I'm really very appreciative of your confidence. Okay, so have a great day, everyone. I'll see you all next week.